Okay, let's get going on nutrition, growth, and metabolism, folks. Okie doke. Let's turn these lights off, could we? Okay, this is lecture 3. Okay, again, here's the key words. This is what we're going to cover today. There are, in the situation with regards to metabolism, which a lot of this lecture is about, as I've mentioned already, metabolism, the biochemical pathways and what have you, there are extra pathways there are in bacteria, and there are certainly much greater diversity in pathways because you're dealing with a kingdom again, not a species. And a lot of the biochemistry you've had is, of course, directed at the biochemistry of human beings. Okay, so you're going to have had some of this before, but the other issue is we're going to look at it more from a different perspective. Unfortunately, we do have to discuss the Krebs cycle, and and many of you, I imagine some of you may have had this 3 or 4 times before. I don't want to do it. It's not from the point of view, though, of you don't have to memorize the formulas of the chemicals involved. It's not what this is all about. What I really want you to just understand, okay, what this is about, so you can answer sort of different questions relate to the understanding of it. So I think it's a little different way you've had things. And it relates specifically also, what we're going to be covering is how things again relate to bacteria. So, take the beginning here. If this was a human being, you've got 5 different terms here. We're all aerobic. By definition, people respire aerobically. If we want to respire anaerobically as we do, the consequences are, of course, that we will respire anaerobically in some circumstances and we die. Okay, we're obligate aerobes, human beings. Bacteria there are 5 different possibilities here. These are general terms. Okay, so again, we're going to have some stuff you've had in biochemistry in this lecture, but it's going to be with a different slant. Okay, so we're going to talk about that, oxygen use. That's the biggest issue in bacteria. We're going to talk about things that are important to bacteria to survive. Metal ion uptake. We're going to talk about how bacteria grow. If you put a human being in a hot spring, I don't know, about 70 degrees centigrade maybe, they probably wouldn't do very well. But many bacteria live in hot springs. There are bacteria can survive in the Antarctic. You wouldn't do too well sitting under an ice, under a glacier or something, unclothed. So basically, bacteria have a lot more versatility, and there are 3 terms that you need to know. And we're going to relate to specifically where the human being and the bacterial. So I gather you had 2 long hours. Anyway, you're okay. We're just going through the general stuff here. Generation time. This is a term that's really almost never used by practicing bacteriologists. Unfortunately, I don't know why. There's a question periodically on the boards, and so that's about the only reason and I give you an example when we go through of how you would answer a question on generation time. Okay? The growth curve is something that a practicing bacteriologist would use and does use all the time. We'll talk about growth curves. Then we're going to get, as I said already, for those of you who just walked in the room, we're going to again be dealing with the Krebs cycle, but it's going to be from a different perspective. It's from the perspective of what do bacteria do when they're stuck. When they're in a situation where they can only grow, they need particular foodstuffs, and they're in a bind basically, because we have a bind. It's simple. We go and eat something, but bacteria may be in a situation where they have no option as to what's available to them, maybe in a particular environment, and so different pathways get turned on and what have you. Okay, so this lecture all about energy metabolism, which is what we primarily get from foodstuffs, metabolism, oxygen requirements, metal ion requirements. Basically, what do we need to do to grow bacteria, and how do we understand how bacteria grow and how they're doing under different circumstances. So to begin, bacterial requirements for growth. Again, oxygen or its absence, because some bacteria do perfectly well in the absence of oxygen. In fact, some bacteria hate oxygen so much Now, I'm sorry, I realise you didn't get a break till 10 o'clock, but I didn't see anybody out in the hallway, so anyway. Okay, so oxygen or its absence. Some bacteria basically just are actually killed by oxygen. And we're going to give these 5 terms that I talked about in a little bit and define them. Energy. Any form of life on this planet needs the energy stored in ATP particularly, and it usually comes from the bonds of NADH or NADPH. And so bacteria, being no different to any other life form on the planet, will generate ATP. And so we're going to talk about, but again, there's these unique situations that I talked about where bacteria are stuck, where they're in an environment where they can't really get what they like to have and what they need. And so they've got pathways that are turned on which you don't see in for example, human cells. So you will have some, and again, for those of you who just walked in, I'm not giving a lecture here. This is microbiology. It's not biochemistry. Pick up any microbiology textbook in the country, and for some unknown reason, most of the chapter is on glycolysis. On the Krebs cycle and glycolysis, which is what we're going to be covering. There are other metabolic pathways. You all know this. There are pathways involved in nucleotide biosynthesis, amino acids, fatty acid metabolism, you name it. It's barely there in any microbiology textbook in the country. Now, part of it is because there's some logic to this, because of the issue of understanding what's unique about bacteria as regards their oxygen requirements and how they use them and why they need them and what have you. Okay, so that's the issue. Is we're going to be dealing with things relating to glycolysis and the Krebs cycle, not because of the fact that other pathways are unimportant, but they're just generally covered more in biochemistry. Okay? So, energy requirements we're going to be talking about. Nutrients we're going to be talking about. Now, there were 3 terms I told you I'm going to give you as regards the temperature that bacteria optimally grow, and pH. So these are issues that relate to how bacteria grow and metabolize. All right, let's start with these 5 terms. Obligate aerobes. Okay, you're an obligate aerobe. I know that most of you, your minds are bigger than a bacterial cell, but you're still an obligate aerobe, whether you like it or not. Okay, grow in the presence of oxygen. There's no fermentation or oxidative phosphorylation. Now, yes, human beings will, for example, produce lactic acid, which is anaerobic. You're probably actually a facultative anaerobe, but let's not get off the subject here. Okay, an obligate aerobe will basically no fermentation. Okay, that's the point, and I'll explain what I mean by that as we go along. But there is oxidative phosphorylation. There has to be oxidative phosphorylation. We have to generate ATP in this situation. An obligate anaerobe is the opposite. An obligate aerobe has to grow in the presence of oxygen. You stick it in the absence of oxygen, it's real simple. It dies. You take an obligate anaerobe and you put it in the presence of oxygen, and it dies. They are the 2 extremes. If you actually grow bacteria in a flask, you will see that an obligate aerobe will sit in the top. You'll see the cloudiness of the bacterial growth at the top of the tube where the air is. If you take an obligate anaerobe, you won't see anything if you put it in the presence of oxygen. So these are real things, and they're used in the clinical microbiology laboratory in a practical sense to how we use, in terms of bacterial identification for the diagnosis and treatment of infectious diseases. And that's why we're going through this. Okay, obligate anaerobe, no oxidative phosphorylation. There is a Krebs cycle in obligate anaerobes, because they do need to carry out metabolism. And as we go into this lecture more, we'll stress the bifunctional nature of the Krebs cycle, being in terms of producing metabolites, but also generating stored energy in the form of ATP. But again, an obligate anaerobe is killed by oxygen. That's what makes it obligate. It has to be. Another term that's sometimes used is strict. They're sometimes referred to as strict anaerobes. And I don't know, those are probably the only 2 words that will be used, but if you see any other version thereof here or anywhere else, you should be asking yourself, what does it mean in terms of, are we talking about an obligate aerobe? Are we talking about an obligate anaerobe? What are we talking about here? What does it mean? They lack certain enzymes. This is one of the reasons they're obligate anaerobes in terms of being killed by oxygen. There are Just part of metabolism, which is beyond the scope of what we're going to get into here, you do often generate nasty things like hydrogen peroxide. We generate free radicals, such as superoxide radicals. Nasty things get produced, and these organisms, generally speaking, aerobic organisms have a way of getting rid of these noxious substances, such as superoxide and hydrogen peroxide. These enzymes are generally lacking in obligate anaerobes. That's why they don't live well in the presence of oxygen, because they're often generated from oxygen. You see, oxygen is generating hydrogen peroxide. And basically, a lot of this stuff actually comes as byproducts of oxidative phosphorylation, which by definition strict anaerobes don't do. Okay, so that's what an obligate anaerobe is. An organism that not only will not grow, not only doesn't carry out aerobic respiration, but is actually worse than that. It's killed by oxygen. We will have a whole lecture on anaerobes that I will be giving in a few days, and we will get more into obligate anaerobes. The reason this will happen is because, as we'll hear in greater detail when I give you that lecture, the obligate anaerobes are normally dumped together. Because if you think about it, you are, as I said before, you're actually, I suppose I said, you're really like a facultative anaerobe, a term we'll deal with in it shortly. But the bottom line is your tissues are full of oxygen. And you can imagine an obligate anaerobe is not going to grow in a regular tissue in a human being if it's full of oxygen. They're going to die. So obligate anaerobic infections are things that occur in situations like diabetes, where there's a cut-off of oxygen to a limb, or some situations that there may have been like a necrotizing myositis situation. There's been some damage so these organisms can grow. And again, we have a whole lecture on that later. And we'll come, so what we say today needs to think about it in that context later. Obligate anaerobes. Another definition, aerotolerant anaerobes. We've said already that what makes an anaerobe is the fact that it doesn't utilize oxygen in its metabolism. But it doesn't have to die. I mean, you can have an organism that could theoretically, and it's not theoretically, it happens, you could have an organism that will respire anaerobically, that is to say, it does not use oxygen, but it might not be killed by oxygen. And Clostridium perfringens is a strict anaerobe, but is somewhat aerotolerant, as an example. And aerotolerant anaerobes respire anaerobically, just like strict anaerobes, but they're not killed by oxygen. They can tolerate oxygen. So that's the difference between, it's obvious the difference between a strict aerobe and a strict anaerobe. The key issue here is the oxygen tolerance of these anaerobes. That's what makes this group different. Now, a facultative anaerobe is an organism which would prefer to grow aerobically. It would prefer to utilize oxygen. If you think in terms of evolution and nature and what have you, that makes perfect sense. Because, as we're going to see shortly, you produce a lot, and many know already, you produce a lot more ATP in aerobic respiration. You store a lot more energy generated from the bonds of the foodstuffs in aerobic respiration than you do anaerobic respiration. So by definition, it would be totally illogical to have an organism that would prefer to grow anaerobically but could grow aerobically. The word facultative means basically they prefer to, they can adapt, they can change to become anaerobic, but they're actually, most of the time, they're aerobic. Okay, so facultative, don't get the impression, just you see the emphasis on the word anaerobe here. The real emphasis is the word facultative. They adapt to, they can grow anaerobically if they have to, but they're really, most of the time, they're going to grow aerobically. And most bacteria are actually facultative anaerobes. Things like E. coli, for example, is a facultative anaerobe. So, facultative anaerobes can carry out both fermentation and aerobic respiration, and yes, they of course will survive in oxygen. In fact, they prefer to grow in oxygen. Okay? So that's our full definition. And these are definitions. I mean, there's some logic. There's logic to it, and I want you to understand the logic. But these are definitions. The microaerophilic is exactly what it says. It's not talking anything about whether the underlying metabolic pathways and what have you, because actually there's precious little known about in this day and age about microaerophilia. But the word is what it says, is what it means. Micro, a little bit. Aerophilic, likes oxygen. Lacks a little bit of oxygen. That's what it says. That's all you need to know. They grow in low oxygen concentrations. They are killed by high oxygen concentrations. If it said macroaerophilic, it wouldn't mean that. Okay, so these are organisms that do well if there's a little bit of oxygen around. They've presumably got enough enzymes around that they can get rid of the metabolic nasty byproducts that we talked about earlier. But if there's a lot of oxygen around, there'll be enough nasty things produced, presumably that it kills them. And I use the word presumably because there's not a lot been done with these things, and we're not going to get into the actual mechanisms. But that's the issue. So it's a definitional term, microaerophilic. And there are certain organisms. Why do we care? Well, we care because there are certain organisms that grow in this fashion. And if you don't know this, then it's real simple. You try and grow them the way you would generally grow things in the clinical microbiology laboratory, and nothing grows. And I will talk about one of the organisms later on in the course. I think it's Campylobacter, but we'll discuss this business about microaerophilia. And in fact, there were situations where for many years we didn't even know that certain diseases even exist because nobody ever thought to grow them under these strange conditions. Okay? So it's not just a theoretical thing. It means something. And it does mean these things are actually translated to, first of all, what you do in the clinical microbiology laboratory, because as you'll hear when we get to the strict anaerobes later, it's a totally different way that you deal with strict anaerobes than you deal with aerobes. It's a piece of cake to grow aerobic bacteria. You put them in a tube, you stick them in an incubator, they grow. Strict anaerobes are a pain in the neck. They're absolutely horrible to deal with, even in this day and age. And so it relates to reality in diagnosis and what you do in a clinical lab. It's real simple. If you think for example, it's very common to see strict anaerobes in a wound. Well, you go ahead and you grow them and the way you would regularly grow aerobic bacteria, and nothing happens. Well, duh. Does that mean they don't have a bacterial infection? Well, of course they have a bacterial infection, but you just didn't know what you were doing. Okay? So it means something, and that's what we're trying to give you the basis of it here. But the practical thing is what has led to the way we do things in the clinical microbiology laboratory. Optimal growth temperature. Again, it's not just an issue of learning this stuff. Oh, that's a mesophile, that's a psychophile, that's a thermophile, that one. No, it means something. We are mesophiles. We basically, human pathogens, by definition, we are 37 degrees centigrade. We're not 38 degrees centigrade. We're not 40 degrees centigrade. We're always 37 degrees centigrade in terms of our body. That means a pathogen that will grow, that's basically been in contact with human beings for millennia, can say, that would be a good temperature to grow at. Okay, most bacteria of interest to human beings are mesophiles. They grow at temperatures close to the human body temperatures. And we will talk about some exceptions, like, for example, M. leprae, the causative agent of leprosy, which grows at about 33 degrees centigrade preferentially, when we get to the lectures on mycobacteria. Psychrophiles, close to freezing. Well, maybe that doesn't mean much, but it does. There are certain organisms that if you put them in a refrigerator, they will survive and grow, whereas most of the other bacteria will die off. So if you stick them in a refrigerator and leave them there for a few days, those organisms will survive. And so what that means is you've enriched for that organism, and you can use it as a way of finding an organism you normally wouldn't be able to find because there's all this other bacteria that are in the sample by just sticking them in a refrigerator for a few days. And that is used in a clinical microbiology lab. Thermophilia, that we probably would never be used in anything to do with clinical microbiology. These are organisms that can survive in hot springs and close to boiling, and this more relates to that horrible term I said I'd never say again, the archaea, which has got nothing to do with human disease, but I've said it anyway. What can I say? I lie. Okay, so thermophilia is the other extreme. So these are the three things. We're mostly interested in thermophiles. That occasionally is of use, and thermophilia doesn't really relate very little to human disease. pH. Most bacteria, surprise, surprise, of interest to human beings anyway, grow at a neutral pH. Why? We're neutral pH, where pH in the human body is about 7.2. Most bacteria of relevance to us, that's where they grow. However, some can survive quite well under acid conditions and some under alkaline conditions, because what happens is there are often metabolic products, which we will talk about shortly, such as short-chain fatty acids in particular, such as lactic acid as an example, succinic acid, acetic acid, propionic acid, what have you. Short-chain acids will be produced and change the pH. And an organism that produces a lot of acidic metabolic products, if it can grow in acid conditions, it's doing itself a favor. And for example, some streptococci, for example, will grow under acid conditions. Okay, so basically that's what we need to know about the range, the possibilities of pH, how they can grow and survive. Nutrient requirements. No big shocks. Carbon, nitrogen, phosphorus, sulfur, and metal ions. And if you haven't had this somewhere before, like even in high school, I'm not going to emphasize it. So that's clearly. But what we're going to talk again, now we're going to get into what's unique about bacteria, some of which you probably have, most of you probably not had before. Some of you may have, who've taken sort of bacteriology at some point or other. Okay, let's first of all talk about siderophores. Certain bacteria such as E. coli and mycobacteria, for example, produce siderophores. Siderophores are basically small molecules that will bind to iron. Iron is an important thing to both human beings and bacteria in terms of certain enzymes need iron as a cofactor. And there are actually enzymes, actually proteins that you find in mammalian tissues and body fluids such as ferritin, and there are other sort of enzymes that are there that are involved in iron binding and scavenging. So the point is, an organism that produces a siderophore is at an advantage in the environment, but it's also at the advantage when it infects a human being, because it means that it can compete with the human being to scavenge for iron. It's got an edge, and indeed it can be a pathogenesis factor, which we'll talk about later also. So what happens is there's a receptor. The siderophore is secreted. It binds the metal ion. It then binds to this receptor and is internalized as a complex. So the siderophore is the iron-binding small molecules, such as enterochelin. They usually have a couple of hydroxyl groups, which sort of kind of like attach to metal ions. Okay, those are siderophores. Okay, now we're going to now talk about how we know that we've actually grown bacteria in the first place. Basically, there's only two ways to grow bacteria. You either grow them on a plate or you grow them in liquid. If you grow them in liquid, those of you have ever seen, I think I'm going to redo this slide for next year. We'll make it a little clearer. Those of you who have ever seen a tube of bacteriological media, before you add bacteria to it, will know that it's a brown, transparent liquid, lightly colored tan or brown transparent liquid usually. But it's not cloudy. It doesn't look like milk. If you get a very, very turbid suspension of a bacterial suspension that's been grown up, it will look like milk. That's turbidity. Okay, so one way you can tell that bacteria are growing is whether you put them in liquid, it changes it, and of course it's a gradual thing. You don't stick them in a tube and bang, they're gone, they're growing. It takes maybe 24 hours or so to grow them up usually, and in case of some organisms it can take weeks. But as you put them in this liquid, this transparent, lightly colored tan or brown liquid, you can measure the amount of turbidity or cloudiness which comes from the bacterial particles that are present. And that's one way of measuring the bacteria are growing. Now, in this instance, you don't know if they're alive or dead, because a spectrophotometer, a device that measures turbidity, couldn't care less whether the bacteria are alive or dead. All it cares about is that there are particles there that produce cloudiness. So if you took a suspension of bacteria and you threw in some penicillin and killed every organism in the tube, the turbidity would not go away. You just have dead bacteria. Okay? So the one way of measuring bacteria is to measure cloudiness, which measures the total amount of bacteria that are there, live and dead. On the other hand, when you grow bacteria on plates, you can measure the viable bacteria there. If you streak a plate, what you'll see is a line where the metals touch the plate. You won't see basically the plate looks basically like it did before you started, except there's a line on the plate where you've touched it with the bacteria that you're trying to get to grow on it. And after 24 hours, individual colonies will grow up, and the individual colonies consist of millions or billions of bacterial cells, making a colony which is visible to the human eye. Now, if there's a lot of bacteria that you actually streaked on the surface of the plate, you'll get a lot of colonies. If there's a few bacteria there, you'll get a few colonies. The issue is that this is measuring the fact that you started with live bacteria, because by definition, if they were dead, you wouldn't get any colonies. You could stick 1 billion dead bacteria onto a culture plate, and of course nothing will grow. So this is a way of measuring viable bacteria. Okay? And we do use these. This is standard microbiology to measure most viable bacteria in some circumstances, and in other circumstances to measure turbidity. And I'm going to take you through a couple of examples, different examples, because I found it's confusing over the years when I cover this separately. So I've made 2 separate slides here to explain what I'm talking about. All right, I'm going to show you the growth curve, first of all. 2 different growth curves. And you will not understand this unless you follow what I said before this slide. If you don't get it, and most of you will, I'm sure, you wouldn't be in medical school if you were stupid, but if you don't get it, for whatever reason, just go home and look at the slides again and think about it, okay? All right, the growth curve. Here we're measuring colony-forming units. Now, we've said already that we're measuring live bacteria. Doesn't matter how you measure bacteria, there are always going to be 4 components to a growth curve. First of all, a lag phase. You don't usually stick a gallon of bacterial cells, onto a culture plate. You put a few cells, a few hundred, a few thousand. So little that initially, basically, well, we'll come back in that's more relevant to the second slide. But the bottom line is, you put a few cells on, and it takes a while before anything happens. There's a lag. Okay, so initially you don't see anything, because the bacteria have got to multiply sufficiently to produce visible colonies that you can see with the naked eye. Okay? So there's a lag phase. Then there's a log phase, and what that means is exactly what it says, logarithmic. They grow really fast. That's the log phase. They're growing logarithmically. There reaches a point. This is a plate you're talking about. They're in a fixed spot on a plate. There's a limited amount of medium and nutrients or whatever around these bacterial colonies. And in this instance, and so anyway, what's going to happen, regardless of what's going on, is the number of colony forming. Actually, this is. I'm going to have to redo all this slide. I'll think about this later. But anyway, let's keep going. So the next stage of the growth curve is you're going to see with colony forming units, you're going to see a stationary phase when the bacteria stop growing. And then there's going to be a death phase. Okay. There's a problem there. I'll fix this later. Okay. Anyway, now let's go to the next. Let's go to turbidity. All right, same thing again. Lag phase. Turbidity. They don't sort of... okay, I know what the problem is. Okay, let me back off a second here. Okay. My mind did a mental flip there, and I started thinking about plates. Okay, let's back off a second here. They'll have to delete this from the part that goes on the net, or we'll look stupid in the entire universe because people read our website. Okay, let's start again. What we're actually doing here with colony-forming units, what we're doing is we're starting with a tube. Okay, this is the issue. Okay, we're starting with a tube. There's a fixed number of bacteria that have been put in the tube. Okay, let's say it was 100 bacterial cells, it was 1,000 bacterial cells. We look in the tube. We don't see anything because you cannot see 100 or 1,000 bacterial cells will not produce enough of turbidity that you'll see anything. Okay? All right, so what happens is at different time periods you will take along the way, if you're measuring colony-forming units, what will happen is you'll take an aliquot, a small portion on the tube, a known volume, and you will streak it on a plate. Okay? So the issue is here, you're not growing the bacteria on plates to start with. They're growing in liquid if you're going to do a growth curve. You'd always do a growth curve by growing them in liquid. Okay? But what you're going to do then is you're going to, you want to know how many live bacteria are there at different points in time. So what you do, initially it's growing in the liquid, nothing happens. The bacteria is just taking a while to grow. And each step along the way, you're measuring on a plate how many viable cells are there present on the plate at each time point over a period of, let's say, 24 hours. Okay. Now, again, there will be very few live bacteria at the beginning. Okay? The cells will grow logarithmically, as I said. There will be a stationary phase when the liquid, the fixed concentration of media is there, and the organisms have used all of the media, and so they will stop growing at this point. Now, what's going to happen? We already talked about the production of things like acids and alkalis and what have you, toxic products of metabolism, superoxide radicals and hydrogen peroxide and what have you. At some point in time, these bacteria just can't take it anymore, and they die. So the total number of bacteria in the tube hasn't gone down, but if you take that portion of the tube and you streak them out, you'll find that you've got decreasing numbers of bacteria as time goes on. Now, the reason I made this slide up was because I'm going to show you the contrast between these two situations, because it is different what you see with colony-forming units versus what you see with turbidity. And if you've most textbooks, like all textbooks I've ever seen, will show you just one of these slides. And I think it's very confusing. So we're separating this out to make it a little clearer. All right, so here we're dealing with colony-forming units. All right? Now, in contrast, when we go over to doing things per turbidity, it's a totally different, it looks very much the same, but what we're measuring is not. And the big not part is this part. Autolysis versus death. Let me go through the whole thing and let's make sure this is clear. All right, again, if you measure, now we've got a tube of liquid, exactly the same things we had in the first place, but here we're not taking aliquots and streaking them on plates and measuring the number of colonies that we're getting. What we're doing is seeing how cloudy is it in a spectrophotometer. We will see, again, the lag phase, the log phase, the stationary phase. Nothing is different. It doesn't make any difference if you measure turbidity. Excuse me. It doesn't matter if you measure turbidity, colony-forming units, or you measure turbidity. You're going to see the same thing. This is where the difference comes in. Some bacteria are autolytic. They self-digest. For example, the pneumococcus is an autolytic organism. The pneumococcus, in fact, it's so autolytic that if you grow up a liter of pneumococcus in the laboratory, you'll come back next morning and there's nothing there. I mean, it kills itself and it digests itself. Now, an organism could die, and lyse, but it could die and not lyse. Okay? Most bacteria are not autolytic. So if you have one curve and you put on it colony-forming units and turbidity and don't understand the difference, you can't work out what's going on. It doesn't mean anything. Okay, so in this instance, all the autolysis means exactly what it says: self-digestion. They chew themselves up and they dissolve. Now, in the case of an autolytic organism, yes, you would see a decrease in turbidity because an organism is instead of having little particles there, it's dissolved. And yes, it would look the same at the death stage because by definition, an organism is autolytic. They are also going to be definitionally, they're dead. But the corollary, the opposite, is not true. You could have an organism that dies, most do, that will die, and will not be autolytic. Okay, so that's what I want you to think about. Look, that's really the issue, is that autolysis is something we're going to be discussing at least twice more in this course as relates to antibiotics, and we're going to be talking about it in pathogenesis, and we're going to talk about it in the pneumococcus lecture, actually probably 3 times. Okay, so know the word autolysis. You'll hear it again. And basically, it's not complicated. I'm sorry I got confused myself just now. But anyway, it isn't complicated. Just sit down and think about the difference between, again, the major issue is this. If you get this part, this is basically, any way you do it, you're going to see this is going to be the same. But it doesn't necessarily mean that death and autolysis are going to correlate. And you could get different results on different curves. And if you ever see a curve like this, you need to basically ask, think about it. There may be a question that relates, for example, to autolysis. Okay? And then so I don't want to hear anybody say, I don't know what autolysis is. I don't understand what you've been told. Okay? All right, so take a look at it. Okay. All right, generation time, which I said already I consider totally unimportant, but it's a term that you need to know. And it does show up on the boards periodically. The time for bacterial mass to double. I'll just give you a simple example here. And so, if 100 bacteria are present at time 0, and I said to you already, you'll generally start with a few bacteria when you're growing something. If the generation time, how long it takes the number of bacteria to double in number is, for example, 2 hours, after 8 hours there will be 100 times 2 to the power 4. Take a look at that slide on your own. make sure you understand it. If it has a different generation time there, if it has a different time period, it starts with a different number at the beginning, you should be able to work out how many bacteria there would be. And that's why I put that example there, so you can go through it on your own. Okay, now then. I should be looking at the clock up there, apparently, because my watch is off. Anyway, so I think I'm going to probably run over about 5 minutes again, maybe. Okay. Sugar catabolism. Now, I've said to those, most of you were in the room at the beginning. Those of you that, well, actually, a few of you got tired, as I am, and got exhausted and left the room for a few minutes to take a break. So, anyway, you weren't here for this part, and I don't blame you. So I'm going to say again that sugar catabolism. We are going to mention the Krebs cycle. I will try and make it clear what part you need to know and what you don't. Okay. It doesn't make much of a difference. When you take your boards, you're going to be learning stuff from biochemistry anyway, so you're going to know every metabolic pathway under the sun ad nauseum anyway. But for the purposes of what I'm doing in this course, let me explain. I will try and go through what I want from you. Okay? All right. Again, bacteria can grow aerobically or anaerobically. We mentioned these other possibilities of microaerophilia and aerotolerant anaerobes. Those two definitions are out the window now for what we're talking about now, because these are functional things. I'm really referring now, focusing on strictly aerobic organisms, strictly anaerobic organisms, or as we say, just anaerobic organisms, and facultative anaerobes. And we're going to talk about the different situations and what happens. Okay? All right. Glycolysis. The Embden-Meyerhoff-Parnas pathway, which, as most bacteria, and certainly also humans and plants, and you've had it before. There are other pathways that, for example, this one is not found in people. But these, and I shan't spend any more time than this on them, except to say there are these two other pathways, the pentose phosphate pathway, which generates, and that's the big deal, NADPH. We need for different metabolic pathways, we need NADH or NADPH. Most of the time you need NADH, but you do need NADPH now and again. So the pentose phosphate pathway is specifically to generate NADPH. And I won't go on to say any more about that pathway. Because and I won't say anything about this pathway either, except to have named it, so you've come across it. And you may not have come across that pathway before, again, because it's only found in a few bacterial species. For the sake of completeness, it's mentioned. But what I'm going to focus on now is glycolysis, the Krebs cycle, and oxidative phosphorylation, and why it means anything to a microbiologist or to a medical student taking a microbiology course. Okay, glycolysis. Okay, again, I'm not focusing on the pathway and all the intermediates between glucose and pyruvate. Many of you have got good memories. You probably still remember it. I haven't had a biochemistry course, so I did a biochemistry degree back in 70 through 73, and I certainly don't remember all the components and don't wish to. Okay, what I do wish you to understand is what's going on. All right, glucose, a 6-carbon sugar glucose, is broken down into pyruvate, a 3-carbon intermediate. That's what glycolysis is. Two things happen during glycolysis. NAD to NADH. We reduce this compound NAD to its reduced form, has a hydrogen attached. At the same time, ADP is converted to ATP. The energy stored in the bonds of the molecules, like glucose in particular, is now transferred into that terminal phosphate. And ADP is converted to ATP. Now, if that was all that happened, it's very simple. You'd have dead bacteria. Because it needs NAD. It needs NAD. It needs NAD to be a terminal acceptor of hydrogen in certain enzymatic reactions. So, if that's the end of the story, well, that's it. You know, we have nothing to discuss. There's no bacterial life on this planet. But no, of course it isn't. Bacteria are smarter than that. Okay, now there's two things that can happen. One is fermentation. Okay, in fermentation, the 3-carbon compound pyruvate, pyruvic acid, is now converted to short-chain alcohols and fatty acids. Now, that's one slide. Okay, we could have, or one part of one slide, we could have a book on this. This is a bacterial kingdom. Maybe we should have a book on it, I don't know. But in this day and age, nobody cares. Okay, that's just the way it is. Nobody cares about metabolism in the year 2006. So we're not going to go into all the possible pathways and all the possible alcohols and fatty acids that could be generated. But if you ever see anywhere else that anybody talks to you about metabolism of any small molecule like acetic acid, lactic acid, etc., you know you're talking, in this situation, you know what you're talking about is fermentation. And you should know enough to know that without everyone being spelled out, because we probably would need a book to spell out every possible product. So as long as you've got the concept, you're in business. Now, at that time, the NADH is converted back to NAD. So the primary purpose of fermentation is not to generate a bunch of short-chain alcohols and fatty acids. It's to convert the NADH back to NAD. Those are the two parts of anaerobic respiration. And that's all this slide says. The summary of the last slide. NAD to NADH, NADH back to NAD, and ATP is generated during the process. That's anaerobic. The Krebs cycle is what an aerobic organism carries out. And I apologize, I'm going to go on a little longer because Gene Mayer is the culprit. You should throw bricks at him because he told me you're all tired and waiting outside and I should wait a few minutes, so I did. Okay. But anyway, regardless. In aerobic respiration, the Krebs cycle, that's taking the place of the second part of fermentation. Again, we have the same process going on here. NAD is converted back to NADH, but we also have in oxidative phosphorylation, the NADH is converted back to NAD. This is what's doing what fermentation does. And the hydrogens are passed to the oxygen and with water, and we get a lot more ATP. Now, what does this mean practically? What it means is you get the actual numbers I'm not putting up here, but there's a hell of a lot more ATP produced from oxidative phosphorylation than there is from fermentation. What that means is strict aerobic organisms with a strict or facultative, aerobes get a lot more energy out of their food than do anaerobic organisms. What that means is that they're going to grow a lot faster and they're going to do a lot better. And that's going to tell you straight away one of the reasons, as we get into our whole anaerobic lecture later, why strict anaerobes are so hard to grow. One of the issues is they got killed already by high oxygen concentrations. But the other issue is the fact that they use a lot less energy. They grow a lot more slowly, and this is the reason. Strict anaerobes don't do this. They will have a Krebs cycle, but they don't have oxidative phosphorylation. So again, just like we said with fermentation, there's two parts. Glycolysis to produce the ATP. We convert the NAD to NADH, and the second part, the Krebs cycle, converts things back again. Now you notice at no point in here did I show any intermediates, any chemical formulas, or anything else. As I say, I'm an easygoing guy. If you want to memorize it, go for it, okay? But I'm not asking you to do that, okay? Now, I will show some of this stuff in a second. You don't need to memorize these formulas. I stress this. I think it's ridiculous, but if you want to do it, do it. Okay, oxidative phosphorylation. Convert oxygen to water, and phosphorylation by definition, converts ADP to ATP, sometimes referred to as the electron transport chain, and it involves ubiquinones and cytochromes as intermediates that will grab free radicals along and transfer electrons along the way. Okay, this gets a little complicated. There's really only a couple of pieces of information here, but I think don't worry about all these names and what have you. You don't need to memorize all these names. What you need to get out of this is what we're going to talk about now is basically 3 or 4 things which I'll explain. First of all, if you're growing bacteria in a fixed situation, such as a test tube, as opposed to being in an environment, if they're in a test tube, there's only a certain amount of stuff there. Or if they were, for example, in the environment, there was a particular situation, let's say there was a glob of fat laying around they were growing on, and there was nothing else. Or there was a hunk of bread laying around, and they were growing on that and nothing else. There were situations where bacteria are limited by the fact they can only metabolize fatty acids or sugars because of where they are and what's going on. Now, they're smart enough, they've developed over the years, that they can handle this. And what happens is that if you just use the Krebs cycle to produce intermediates, what's going to happen is that if I pull off oxaloacetic acid or fumarate or malate for something else, such as to make nucleotides or amino acids, you don't have the components of the Krebs cycle. So the Krebs cycle is bifunctional. It's got two purposes. It's to break down pyruvate. There's two carbon dioxides come off here. It's to break down this molecule and totally degrade it. Well, actually, there's one carbon dioxide there, one there, and one there. It's a three-carbon compound. This pyruvate is totally degraded. And so, well, that's what bacteria do. The function of the Krebs cycle is to totally degrade pyruvate. No, of course not. Of course not. The function is to produce NADH, which can then be shoved into oxidative phosphorylation to generate ATP. All right. So if we do. So during this process, this pyruvate derived from a sugar, is basically going to be totally destroyed. If we want to produce other metabolites, this is not good enough. So we're going to talk about two situations here. This will be the end of the lecture. There'll be about 2 or 3 slides now. Sorry, I know it's 3 hours. This is right at the end. This is probably the most confusing part of the lecture. So wake up, smack yourself on the cheeks, do whatever you've got to do to wake up. Okay, so basically we're talking about two situations, whether the bacteria grow on sugars as their primary source, or if they're growing on fatty acids, and there's not much else there. Now, in this instance, what happens is the pyruvate going through the Krebs cycle, as I said already, would be broken down into carbon dioxide and would feed into the Krebs cycle and would be totally destroyed when the other two carbon dioxides are there. If you pull off, for example, oxaloacetic acid to make aspartic acid, as this cross says, it's gone. Okay, it's gone bye-bye, it's dead, finished. So what you want to do if you want to carry out metabolism when sugars are the sole carbon source generating pyruvate, you want to find a way to replenish the Krebs cycle. And in this case, the 3-carbon compound pyruvate is converted into the 4-carbon compound oxaloacetic acid. This is an extra enzymatic reaction which is turned on when bacteria need it when they're grown on sugars as the sole carbon source. So there may be questions that relate to understanding the process. You're not required to memorize the Krebs cycle. I probably won't ask things like, how do you convert oxaloacetate to citrate? What I will ask you, may well ask you, relates to understanding this process. Okay? Again, this is put here not to make things confusing and give you lots to memorize. It's again now to explain what happens in a situation with fatty acids. Totally different situation. We're using the Krebs cycle again. In this instance, fatty acids, they can be as many as 20, 30 fatty carbons in length, will be broken down into the 2-carbon acetic acid. And it will also feed into the Krebs cycle, and just like the pyruvate, it gets totally degraded. So there's nothing different that I've told you. That's the Krebs cycle. And that's nothing different there either. The aspartic acid, for example, just as an example, generating amino acids in this instance will deplete the oxaloacetic acid. You could deplete the malate, the succinic acid, and everything else for that matter, depending on what pathway you're talking about. But regardless, if they're growing on fatty acids, they have to be able to replenish the components of the Krebs cycle. And the way this is done, in this instance, is this would be coming from the Krebs cycle. We lose these 2 carbon dioxide that I've already mentioned several times as we go from the 6-carbon isocitrate to succinate. But in this instance, what happens is you generate 2 compounds, glyoxylate and malate. Whatever amount of isocitrate you started with, you want to call it X, whatever amount you started with, you end up with 2X as regards the number of molecules at this stage of the game. I will show you on the next slide that glyoxylate and acetate both get converted to a component of the Krebs cycle. So you can pull off half of your Krebs cycle components in this situation and use them because you've replenished it. So this is a unique enzymatic reaction which occurs specifically when an organism is grown under sole carbon source being fatty acids. This slide is meant to show you what we mean by the glyoxylate cycle versus the Krebs cycle. The Krebs cycle is in red. If you look at this carefully, you'll see that almost everything that's in the glyoxylate cycle, the purple cycle, is also in the Krebs cycle, except for 2 things, the things I just showed you, enzymatic reaction 1 and enzymatic reaction 2. The isocitrate gets bypasses. This is where the two carbon dioxides would be lost as the molecules decompose. It bypasses that, and the isocitrate is converted to the two-carbon molecule glyoxylate, but it's also converted to the four-carbon molecule succinate. This whizzes around the Krebs cycle and stays there. Here it grabs another acetate and gets produced a malate. So we get malate and succinate. We double the amount of Krebs components by using this enzymatic reaction. Okay? The punchline. Okay? And don't just learn the punchline. Okay? Understand where the punchline came from. Krebs cycle. Biosynthetic and energy storage, both. You can get, intermediates must be replenished. There are unique enzymatic replenishment pathways for sugars and fatty acids. And that's what's different. This part of it is different about what some of you may have had it, but you don't generally get this in a biochemistry course that doesn't emphasize bacteriology. So that's what's different. And we talk about the glyoxylate pathway a lot in bacteriology because it's unique. And we also talk about the unique enzymes that are used to, again, when bacteria are grown, sole carbon sources and sugars and fatty acids. Okay, sorry about I ran over a bit there. I'm going to make sure my watch, if it agrees with this clock, to